Okay, hello everybody. Good morning and welcome to the last day of SC21. This is the panel number 127. The title is HPC Response to the COVID-19 Pandemic. The chairs today is myself, Piotr Luszczek, and Hatem El-Tayeb. I'm from University of Tennessee and Hatem is from KAUST. Today, we originally had 6 panelists. So we have David Keyes from KAUST in Saudi Arabia. We have Rick Stevens from Argonne National Lab in the US. Unfortunately, Florent Duchesne from Surfax and Friends could not make it. We also have Makoto Tsukabura from Kobe University, Japan, and we online, since they had the conflicts virtually, we have Dan Jakobson from Oak Ridge National Lab in the US, and we also have online Patricia Kovac from the Icahn School of Medicine and Mount Sinai. So this panel is it was started at the SCIEM CSC 21 mini symposium. And by the way, if you'd like to watch this online, I posted the link. The video and the feed from the presenters will be accessible through this QR code. So we had the symposium in SCIEM virtually, and there were some changes since the symposium. So we put this panel together and invited some of the mini symposium speakers to be with us today. So the advances include the recent CFD work that we'll hear about, molecular dynamics for drug discovery, and cancer genetics. Again, these were all will be included in the panel. Another item of interest that you should probably look at the in SC21 program and on the Hub platform is the a new entry for the ACM Gordon Bell Prize. It's a special prize for the HPC COVID-19 research. And so if you look at the different entries there, I just provided the highlights from these entries. There's some work on the droplet aerosol infection. One of our panelists actually will be talking about that. We have language models for the encoding of the SARS-CoV-2 virus. We have real-time response and decision support for the pandemic, and virtual screening. And replication transcription machinery experiment. So these were all entries in the special Gordon Bell Prize Award participants. So what we did for our panelists, we gave them a list of items that and asked them to score how HPC in particular did on this particular 11 items. So probably not all of them will take a kind of response to every single one of them, but we provided a few of them just to kind of give everybody a chance to give their opinion. So we have visualization, we have number 2 molecular dynamics, number 3 fluid dynamics and transport. We have number 4 diagnostics, and number 5 drug selection and repurposing, and number 6 tracking spread and mutation rate, number 7 the forecasting the spread, and number 8 the optimizing the response, number 9 vaccination logistics, number 10 social information and misinformation and how it spreads on the social platforms, and 11 the economical consequences of the pandemic. So without further ado, let me stop sharing and start with David.
Thank you, Piotr. And I trust that this can now be seen. Good. So one of the best ways to look forward is to look backwards for some perspective. So I like to start with Daniel Bernoulli in Paris in 1760, who basically, in addressing smallpox, said, "I simply wish that in a matter which so closely concerns the well-being of the human race, that no decision shall be made without all the knowledge that a little analysis and calculation can provide. Now, this was a part of an address to the Paris National Academy of Sciences, an attempt at a new analysis on the mortality caused by smallpox and of the advantages of inoculation to prevent it. Several remarkable things about this paper. He introduced what we now call the SEIR equation for well-mixed epidemiology dynamics. If you rewrite that equation in modern terms, you'll see a logistic equation for the susceptibles, and it's coupled to an equation for the survivors. This paper actually launched applied differential equations. I mean, it was being, pioneered as a way of doing math modeling in applications right at this time. And in a similar way, I think the COVID pandemic has accelerated many aspects of HPC. Another interesting feature here is that this paper took 6 years to get published, and that's something we've overcome with sort of 1 week preprints in the pandemic period. If we fast forward 260 years minus a few weeks to March 2020, we see the COVID-19 HP Consortium established in the US and the COVID-19 Fast Track established by PRACE in the EU. By another month, Fugaku is brought into service a year ahead of the planned schedule by RIKEN and MEXT expressly to fight COVID-19. In June of 2020, in the first virtual offering of SC21 Digital, we had a special section immediately following the top 500, which traditionally begins that conference on the COVID pandemic, HPC, and the way AI is addressing it. And we had those 3 campaigns, each represented by Satoshi Matsuoka, Peter Coveney, and Rick Stevens. HPC Wire picked that up with remarks on the application of off-the-shelf CFD for spreading Gibbs-free energy estimates for molecular docking, and the use of AI in screening repurposing of drugs. So these came very early on in the pandemic, and of course we'll have updates on several of them today. I think it's fair to say that in June of 2020, if you sampled the floor, the virtual floor of ISC, you would have heard things like, COVID-19 has had considerable positive impact on CSE research, but not vice versa. COVID-19 understanding and mitigation is still essentially a wet lab and clinical story. And where has CSE had an impact beyond research frontiers in actual policy support, diagnosis, or therapy? So by September, at the SIAM CSE mini symposium, our panel chairs today put forward, wanted to come back to this question: how far has HPC actually come in mitigating the pandemic as opposed to being driven by it in the development of tools? And they made a rather bold claim. Actually, this claim was made at time of mini-symposium submission. When the mini-symposium actually ran, I gave this report card on 8 of the 11 topics that Piotr just listed. By that time, I thought visualization had surpassed what we can do with electron microscopy. Molecular dynamics had elucidated the spike protein mechanism, its opening and closing, and when it opened, the ratcheting of that sort of kickstand support to keep it open. Fluid dynamical transport had already been quite successful on Fugaku and in other campaigns to estimate the spread of aerosols. Diagnosis of CT scans of lungs was already at human accuracy for detecting the presence of COVID. On the other hand, for the last 4 topics, I gave it an incomplete. Drug repurposing, a lot was underway, but we hadn't yet moved out of clinical trials into certified drugs. Tracking spread and variants, there was a lot going on, refining the SEIR equation into agent-based models. But forecasting spread had not yet really moved into planning resources or optimizing response or setting policy. So by this time, in November 2021, I'm not necessarily an easy grader, but I do think that we have advanced to a actual deliverable situation in all 8 of these categories, and some of the major players are in fact just about to take over from the mic here, and you will see why I feel this way. So, summary, CSE has been very good for COVID, allowed scientists to visualize 100 million atom biomolecules in greater detail than any imaging system, allowed them to understand the attack configuration, allowed health and safety experts to understand convective and diffusive transport of the virus, gave clinicians new AI-assisted CT scan interpretations, identified hundreds of potential new drugs or drugs to be repurposed, and put them into experimental pipelines in a highly vetted way and into clinical trials, created a lot of computational infrastructure and workflows that will be relevant for future pandemics, and enabled public policymakers to dial the balance between losing lives or suspending livelihoods. On the other hand, COVID has been very good for CSE and HPC. It pushed the #1 ranked system into broad use a year ahead of schedule, created new international HPC consortia, led to sharing of data on an unprecedented scale, created new computer-to-laboratory pipelines. You'll hear about one of those from the next speaker. Focused new attention on the role of preprint archives, accelerated the injection of AI into scientific and clinical workforce, accelerated the development of some new algorithms in MD to skip over unproductive stretches of time and look for those rare events, accelerated the development of new models in epidemiology that were population stratified beyond the classical SEIR and began to take into account geographical aspects, and brought billions of readers and viewers right to the precipice of computational modeling and machine learning, looking in from the public, from popular media accounts, and I believe inspiring many future scientists. And as was just mentioned by Piotr, a new Gordon Bell Prize has been instigated. I won't reread these titles here. It was a very exciting time on Wednesday to attend these talks, and one of the speakers actually won this Gordon Bell Prize and will tell us about the CFD aerosol modeling. Of course, this was the second instance of the special Gordon Bell Prize. 1 year ago this week, it was taken by the team led by Rami Amaro at San Diego, a very multi-institutional team with many scientific merits and many infrastructure merits spilling over into HPC in terms of new versions of NAMD, new algorithmic strategies in MD, and so forth. And these are just some pictures already a year ago of what was happening in terms of understanding the dynamics of that complex body of several billion atoms. And now this year, of course, we just heard yesterday at the award ceremony that a team with Makoto took the prize, and so we'll hear more about those interactions. My second last slide is that AI has become a vital part of HPC and CSE during this campaign, allowing MD modelers to fast forward, allowing expensive simulation kernels to be replaced with inexpensive input-output functional maps, leading to improved throughput, improving on human accuracy, including physician expert accuracy, shrinking false positives and false negatives in clinical tests, and allowing most likely candidates to be identified out of literally billions of small molecules after being trained on a small sample and extended to the large lot to vet things for more detailed model-based simulation. And my last slide is a humorous remark on the effect of the pandemic and the HPC success on science and engineering generally. This is from a combustion colleague at KAUST who observed that when we closed the labs, his lab was useless and many of his students got converted from experimentalists to simulation people. So this has more than doubled the use of CFD simulations, and we do not see this change back to experiments anytime soon. I look forward to my colleagues. Thank you.
Thank you very much, David. Okay, so let's now move on to Rick. You're next.
Hey, thank you. Very nice start, David. Can you see my slides?
Yes, we can.
Okay, so following on David's great introduction to the overview, I'm going to talk about what happened on our side applying HPC and AI to virtual screening and drug discovery. So before I get started, I want to say this was a huge effort with people from across the US and in Europe that worked together in very rapidly assembled teams in February of 2020. I especially want to call out Ian Foster, Arvind Ramanathan, Tom Breton, Austin Clyde, and then and Marty Head, who you heard about earlier this week when she gave a talk. But literally there were over 100 people involved in this effort. So the primary thing that we set out to do was to search for new drugs and also to deeply search for repurposing candidates. And to do that, we needed to assemble very large collections of molecular data and then devise a virtual screening system, and at the same time create basically handcrafted receptor models of all of the plausible drug targets in COVID. And I'll walk you through quickly how we did that. The primary thing that became clear at the very beginning is that the computational throughput for applying classical biomechanics type simulations to do virtual screening was not going to scale to the many billions of molecules that we wanted to screen over and over again. And at the end of the day, we ended up screening about 800 billion combinations of molecules and receptors. And so, so we needed to find a way to accelerate that. And the way that we decided to pursue that was to build AI surrogates. And that's essentially the story here. Our plan of attack, which I'll show you in a second, so we started out by creating a sampling on the order of 1 million compounds, diversity sampled. We then used biomechanical models to dock these from several different docking programs, took that data as training data, trained deep neural networks to predict docking scores based on that training data, and then did inference across the billions of molecules that we had assembled, scooped up the high scoring hits, and then went back and did biophysical modeling on those and deeper theoretical modeling, and then advanced those candidates into the experimental pipelines. So this is, I won't go through the workflow in detail, but on the left here, number 1 is a list of the proteins, the mature protein products that come out of the COVID genome. And there's about a dozen or so plausible drug targets. Some are form complexes, some are not easily druggable, but we prioritize those. We tried to balance across the life cycle of the virus, so we had candidates that would interrupt virus entry, virus replication, virus maturity, viron maturity, etc. We devised this pipeline. We ran this on the supercomputers at Argonne, at Oak Ridge, at TAC, at San Diego, in Europe. So we had lots of cycles. We ran this on all the drug targets for which we built receptors, over 100 receptor models. So each plausible target has multiple receptor models. The output of this ended up being over 2,000 molecules that we either synthesized or procured, and then ran those through functional and antiviral tests, then did a series of secondary screens on close variants of those molecules to get at structure-activity relationships, and then pushed that whole collection of targets and hits forward into various downstream development. So what does a receptor look like? Here's an example. This is from the NSP15, which is an endoribonuclease molecule. It's normally in the functional form is a hexamer, so 6 of these subgroups form into a structure. The blue zone here is one of the potential binding sites. This is a model. We call this a receptor model. And the idea is that you're going to dock small molecules into that blue region and find those that bind well and have low or have high binding affinity, and then use that as a filter across the compound. So for every receptor, you have to do a separate search. Now, the end result of the computational effort on this would be a list, like up here on the left. It would be a molecular ID and then an estimated docking score, which might correspond roughly to a binding affinity. We typically did the biophysical simulations for something like this using OpenEye's tool set and Autodoc Vina's tool set. Some of those ran on CPU, some of those were GPU accelerated. The typical throughput that we'd achieve for biophysical modeling is about 1 receptor drug experiment per second per CPU. And you can do the math on how long it would take if you had 100 receptors and say a database of 4 billion molecules. On average, we would keep only the tiniest fraction of the high-scoring hits as we proceeded from one stage to the next. Early on, we achieved an acceleration of this surrogate, this AI surrogate, of about 50,000x. So we could go 50,000 molecules per second on inference mode. Since then, by partnering with our AI hardware partners that are part of the Argonne ALCF AI testbed, we've been able to push that 50,000x to over 20 million times faster. So the current record for this, running on the Grok hardware, is a 20 million x acceleration over the biophysical models, and that portends, well for the future. It means that you could screen literally billions of molecules in a few minutes without any problem going forward. That's the slide here. So in order to do this, we had to build a very large database. We aggregated data from over 20 different public sources to create a combined data set of over 4 billion molecules and made this all publicly available. We had to also compute properties of these molecules so that we could use that in the deep learning models. And we did a lot of experimentation on which forms of representation were actually best for driving the surrogate. Now, of course, the biophysical model is working on the atomic coordinates and is doing everything with physical force fields and so forth. But for the deep learning, it turns out that images work quite well. We also did drug descriptors, smile strings, and fingerprints, and so on. But here's an example of images. So basically a 2D image rendered from the molecule, and then using a ResNet-like structure to predict the binding energy. And this worked incredibly well, as accurate as any of the other methods. It also had the advantage that we could use attention maps to understand what it was the deep learning was looking at in that molecule to make its decision. And so that was quite useful. The status update. So now about a year and a half since we started this effort, we've done over 800 billion what I'll call compu screens, some combination of biophysical and AI screens. We've experimentally screened over 2,000 compounds on over a dozen drug targets. We have 63 pretty good hits that are in downstream experimental pipelines, several of which are getting to clinical level activity. We've also run 600 additional molecules to actually bracket those original 63 hits to get at the structure-activity relationships. And this large compound database we created is now actually in use in other drug development projects, some related to SARS, some related to cancer, some related to other things. There was a very nice summary of this overall National Virtual Biotechnology Lab effort that Marty Head gave earlier this week in the conference, and I think that recording is probably available if you're interested. Thank you. I think that's it for me.
Thank you very much, Ray. So, Makata, you are next.
Okay, I'm going to share my slides. Okay, what we are conducting now is capturing the transmissions of COVID-19 virus by using fog-fluid dynamics. I'd like to point out the difficulty in the COVID-19 transmissions. Firstly, it is a sort of the emerging infection disease rapidly spread all over the world. So at the initial stage of the pandemic, the scientific data was quite less under the very strict lockdown. And secondly, the reasons of the transmission for the COVID-19 is inhalations of dense aerosols emitted from an infected person at a relatively close distance. So in that sense, that this is a sort of the intermediate between the airborne transmissions and droplet transmission. So the past some of the theoretical ways of evaluating the risk does not work. So we need some more the reliable method to evaluate its infection risk. And anyways, our idea is utilizing the massively parallel resources for terms of the virtual infection risk assessment and the proposal of each countermeasures. Here, what we realized is shown. It is a sort of the digital transformations of the aerosol infection risk for three respects. One is a very close distance evaluations, the effect of masks or the face shield and so on. And the second one is the infection risk in the commuter trains or the taxis and so on. For this, from the CAD data, we developed very fast mesh generation systems and realized sort of the digital transformations by reducing the pre-processing time from some weeks down to minutes. And third one is indoor environment. In that case, usually there is the shape data for the very old buildings or old rooms. So we need a sort of the three-dimensional laser scanning and making the geometries and evaluating its infection risk. And the achievement of our research is we have conducted over the 1,000 infection risk assessments for these 17 months for the various situations shown here, and we rapidly respond to the situations of the COVID-19, which varies day by day. And update from the previous research is now we are conducting a sort of the quantitative risk assessment simulation for policy making. Our past simulation, we just evaluate the risk between the two persons who are on the table or something like that. But now we are trying to evaluate the risk in the whole room, including the very detailed air conditioning system, and as well as some fans or something like that. For example, this animation showed the two situations of the very small Japanese-style restaurants where the 16 persons are sitting there and speaking loudly with alcohol or something like that. And on your left is cases that we just switch on the mechanical ventilations on the ceiling. And on your right is a case we additional ventilations by the kitchen duct and air conditioning systems. As you see that the dispersions of the droplets is quite different. And seemingly, right animation shows more dangerous than the left cases, but that is not true. We can easily evaluate its infections by directly counting the particles reaching to each person from each person. So then we can make a sort of the infection risk matrix in this room by indexing the numbers of the sitting seats from 1 to 16. Okay, and here on your left, this is the cases of only the ceiling ventilations. Here, the vertical number indicates the person of infected person's ID. And on your right is how these infected persons infect to the persons around here. So that this sort of the matrix can be generated from the numerical simulations by counting the droplets reaching there. Interesting thing is that for the cases of only the ventilations on the ceilings, the risk is more or less very condensed. Like the cases only on the desk around here, 1, 2, 3, 4. Here you see, and this is for the desk B here, and this risk is around the counter. But once you switch on the kitchen duct air conditioning systems, you see that the risks are going to be very diverged in the room. Seemingly, light situations seems to be more dangerous, but just taking the average of these probabilities, then you can realize that infection risk can be reduced by half by switching on the kitchen duct and the air conditioning system. The point is that locally, the infection the risk is going to be increased, but taking the average, the risk is going to be half. So I think that this sort of this evaluation is going to be quite important to make the policy to make the policies by the government. And here, by conducting so many simulations with different situations, like only the kitchen duct on, only the air conditioning on, or that setting the partitions on the desk or something like that, or the fast seating or something like that, then you can quantitatively evaluate the infection risks for each countermeasures. Like this is the original cases. The color indicates just how the disease is just COVID-19 and it's the strength of the infections by changing some parameters. And the blue is a Delta variant, and the gray is the original variant. And only the ventilation, so its risk is quite high, but just switch on the kitchen duct, you can reduce the risks by 10% or so. And you can switch on the air conditioning system and the kitchen duct, the risk can be going to be the 1/3. Together with the partitions, then the risk can be half or something like that. So these types of quantitative evaluation is indispensable to proper decision makings. Actually, we have provided these data to the cabinet secretary in Japan, and they utilize this data for their decision makings in Japan. Currently, we are now going to integrate sort of the different simulations together to make the infection risks, the evaluations more precise. Here, on your left, second from the left is simulations of the droplet aerosol dispersions, and we are now coupling these simulations with numerical respiratory tract, and investigating where the droplets are going to be attached on the surface of the airway. And then also we are measuring how the infection risk, how the droplet virus is going to be increased after attaching that on the surface of the airways by solving the very simple biomolecular equations shown here. Of course, we need some sort of the tighter collaborations with the experimental guides to determine the parameter of these simple mathematical models. But anyway, by coupling these simulations, the accuracy of the evaluation is going to be higher and higher. Next simulation is currently underway. We are going to conduct the cases of the with or without masks, and then we are track where the droplets are going to be attached on the surface of the respiratory tract. Usually the attachment is strongly depending on the size of the droplets, and the smaller one or larger one are easily attached on the surface of a nasal cavity, or the size is around 100 micrometer is going to go deep into the respiratory tract. And after attaching, we solve this simple bioregulation equations. It's a sort of the host cell dynamics and how it is going to be increased. Then count the total number of the droplets. This one shows the numerical results showing the viral load against the time, the days. You see that the virus is going to be increased, and it's going to reach the peak after 5 days of infections, and going to be decreased. And looking at that, and here the red line indicates the PCR detection limit. So numerically, you can identify that the effect of the PCR detections, where we should conduct that for the infected person, or something like that. Okay, finally, I'd like to show this. My own evaluation of each method. For the visualization, it really works in our simulations. By just visualizing our results, we can draw much attention from the Japanese peoples, Japanese society, and many people sort of the social awareness can be realized in our simulation. But for the simulation result itself, still it can work. So for the decision making, the policy making, but still there are so much rules for the improvement, especially in the context of the accuracy of the simulations, because the simulation result is strongly depending on the boundary conditions, and especially especially the information of biomechanics, like how many virals are included in each droplet. We still need much rules to improve. Such cases about molecular dynamics or something like that, that is not my field, but still I think it relatively works well, but it's less depending on the boundary conditions. And for the cases of a much more societal simulations, like investigating the number of the predicting the number of the infected person or something like that, it's a sort of the intermediate region. So we should cross. So we should communicate. They should communicate with other fields and combine the information. Then maybe the results is going to be improved. Okay, thank you very much.
Thank you very much, Makoto. Okay, Dan, you are next.
All right, great.
Yes.
Hopefully you can see a slide about now.
Yes, we see the slide.
Awesome. Okay, today I'd like to run you through how we're really confronting a changing world that's leading to COVID-19 with supercomputing and systems biology. We're increasingly really viewing these sorts of interactions in a One Health framework. So the basics of which is that we are affecting our ecosystem, and in turn our ecosystem is affecting us, and pandemics like COVID-19 are simply part of that feedback loop. Our view of how to approach these sorts of problems are holistic approaches, where we're looking at many layers of information, omics information, environmental information across multiple organisms, be that human or model organisms, or organisms in the ecosystem or agrosystem like plants and microbes, and running all those different layers of information through an ensemble of algorithms, whether that be AI, explainable AI, data analytic approaches, to try to get at the combinatorial complexity of the underlying biology. The combinatorics of epistasis in a single human genome is on the order of 10 to the 170. So supercomputing alone doesn't solve that. It's a combination of clever algorithm design with supercomputing that can approach these sorts of problems. In capturing those relationships, we often model them as multiplex networks, and then use the development of lots of network mining algorithms to find the underlying biological mechanisms. And of course, with high throughput structural protein modeling, we can start to interrogate that 3D interactome, be it between proteins or proteins and molecules themselves, or as Rick has described, between proteins and putative drugs. All of that is really to get at conceptual models and mechanistic understanding of these systems that we can be proactive and reactive to solve these sorts of problems. So this is just an illustration of the process we're using explainable AI approaches to find these higher order combinatorial interactions of the omics layers themselves, within and now across all of those omics layers, and how that's all conditional on the surrounding environment. And from that derive more mechanistic understanding. We're doing exactly the same style of approach with clinical information. So with electronic health records, we're fortunate to be part of the DOE VA collaboration, where we have the clinical records for 23 million people going back 20 years here at Oak Ridge. And using these sorts of approaches, we're finding fascinating patterns in the clinical data that, when we combine with the mechanistic omics view, can lead to drug repurposing directly out of the mechanisms we're finding and the clinical data. So this has really led us to a whole range of activities in COVID-19. In some of our early work, we discovered new mechanisms for pathogenesis in COVID-19. that led to a number of clinical trials of existing drugs. It led us really to the conclusion that this is a systemic disease that wasn't just affecting the respiratory system, but affects components of the body all over the body, many, many different organ systems. And in fact, it's a vasculitis. So once it's in the circulatory system, it's almost by definition going to affect a lot of different areas. We're also putting a lot of effort into viral evolution, both of coronaviruses in general, as well as the evolution of SARS-CoV-2 over the course of the pandemic, and being able to understand the mechanisms of evolution leading to these variants of concern. And we have strong evidence inferring that the variants of concern are really emerging not out of serial mutations, but out of recombination of different strains that are colliding in individuals. It's worth noting that a single cell is going to generate probably hundreds of millions of virions, and a single infected individual is going to generate hundreds of billions of viral particles. So if you have two strains in the same individuals, the number of opportunities for recombination and admixture of the genetic material is very strong, very high. And we're seeing actually recombination events in the sort of networks we're deriving of evolution that would explain how these variants of concern pop up. In contrast, if you attribute variants of concern to serial mutation, one mutation at a time, simply building up, the chances of that happening, leading to variants like alpha and delta, are roughly 1 in a quadrillion, 10 to the minus 15. So what the virus is doing is actually parallel processing. It's creating mutations all over the world, and as they bring them together, all of a sudden you have a big leap in genetic load. Now, we're not just concerned about COVID-19. We really never want to go through this again. Unfortunately, the time periods between zoonotic spillover events is increasing. In the 20th century, you would see these sorts of outbreaks roughly a decade apart. Now we're seeing them about 18 months apart. So that acceleration is of grave concern. And we have, with our collaborators, this sort of Swiss cheese model of zoonotic spillover. There are all these barriers to entry, effectively, of how a pathogen transitions from its natural zoonotic host and eventually over to humans, and then spreading in the human population. You can break down this model into components involved in the environment affecting and the zoonotic biology itself, into elements of viral evolution, and then, of course, elements of human behavior and human susceptibility. That transitions our integrative models of how we approach this into broader and broader scopes. So now we're not just interested in the organisms themselves, we're interested in that broadly surrounding environment having to do with land use change and climate change, environmental change, and changing those relationships. And also human behavior and even economics that drives land use change and human behavior. The more shots on goal, the more integration events you have between wildlife and humans, and the more stress you put those wildlife under, the higher your probability of deriving zoonotic spillover events and therefore future pandemics. So I'm just going to touch briefly on some of the work we're doing in the environment. We're really interested in finding the environmental relationships and how those are changing over time. So here we're taking data sets with 14 different layers of environmental information for every point of land on the planet. We're using a creative bit encoding scheme to put those into binary vectors. Each of those vectors is about 500,000 elements long. And then we're doing all against all comparisons, both two-way and three-way comparisons for every point of land on the planet. We're doing this for the past 60 years agglomeratively, as well as in 10-year sliding windows, so we can see how things are in fact changing over time. We're using the DUO algorithm for this, which has been highly accelerated on GPUs that we're running both on the Summit system here at Oak Ridge, as well as the Joules Booster at the Jülich Supercomputer Center. And on the new Joules Booster, we are able to hit 9.3 exaops of performance Overall, this was a 160 zeta op calculation. We think that's about the third largest calculation done to date. To our knowledge, that's the fastest calculation done to date. And the resulting network representing all these relationships, unthresholded, was over 10 to the 17 edges. We did, of course, stringent thresholding to discover the underlying relevant topology. Now, the benefit of doing this with a sliding window is then we can see how that network topology, how these environmental relationships are changing over time and project them onto a geospatial representation of the Earth. Here, blue is representing a stable environment, not many changes in how that area is related to the rest of the world. Orange is our hotspots of change, and you can see how it is pulsing over time. You get pulses of these changes in relationships over the past 60 years. And hotspot areas of concern for things like Hendra virus in Eastern Australia, in Bangladesh and India, Zika in Brazil, Ebola in Central Africa, are all areas of concern. Of course, the Arctic is another big area of concern. Over 30% of our recalcitrant carbon is locked up in peat bogs under the permafrost. As those are thawing, it's clear that methanogens are being enriched for in converting that carbon into methane in many instances. My last visit to the Arctic Research Station in Abisko, looking at these microbiomes, was very sobering. So we're worried about even higher acceleration. That's just a flavor of the different areas we're working in on this. So far, roughly in the past year in our COVID-19 work, has generated 21 manuscripts. 13 of those are published. 9 more are working their way through the review system in a whole range of different areas. We were fortunate and somewhat amused as this work was highlighted in congressional hearings in SST last year, and last year's HPC Wire Award, as well as Secretary of Energy Award. Big shout outs, of course, to OLCF and Bronson for all access to the summit, and Thomas and friends at ULIC for the wonderful access to Jules Wooster and Jack Wells at NVIDIA for helping broker the relationship with ULIC. Of course, all the phenomenal funding sources that supported this work in a variety of ways, and this amazing team of students, postdocs, and staff members I'm so fortunate to lead. And with that, we can take questions, I guess, in the panel.
Yes, thank you, Dan. So now I will be playing the video that Patricia provided. And in order to do that, I actually am required to leave Zoom. So don't worry, I'll be gone, but I'll rejoin in just a second.
Hello, I am Patricia Kovach, the Dean for Scientific Computing and Data at the Icahn School of Medicine at Mount Sinai. I'd like to thank the organizers for inviting me to speak today. Just to give you a little sense about me, I founded the Scientific Computing and Data Group at Mount Sinai in 2011. So I've been here about 10 years. I oversee the high performance computing, the clinical data warehouse for research, and other research databases. I work at the Mount Sinai Health System in New York City. We have hospitals located throughout the city, and we're one of the largest employers in New York City. We have over 8 million patients, around $8 billion a year in revenue. The mission for the Scientific Computing and Data Group at Mount Sinai is to enable and accelerate biomedical discovery through collaborative research using high-performance computing, biomedical informatics, and data science. And of course, during COVID, having these resources available, both data and computational high-performance computing, were really important for researchers and clinicians to help to try to figure out what was going on with COVID. And I'll give some examples of that in this talk. Just some high-level examples, but I'll give you some examples. So I'll just start off with a couple slides on the computational and data ecosystem that we have that was used for for this work. We have an ecosystem where we have some yesterday's medical records being deposited into a data warehouse that's actually sitting in our high performance computing infrastructure. We have a data commons that has both publicly available data sets and Sinai specific data sets available. We have images in there, we have EKGs, we have a blood serum biobank, and we just have a whole rich array of data sets available for researchers to do multimodal data analysis on. So this gives you a little bit of an overview of that infrastructure, and just a little bit more about the high performance computing infrastructure. We have our private cloud. Our high performance computing infrastructure has about 25,000 cores, over 200 terabytes of memory, and about 50 petabytes of raw storage. I'll say that we got an NIH award this year, specifically to help work on COVID and translational science projects, and we got 55 of these 1.5 terabyte large shared memory nodes, and some additional, and 17 petabytes of storage to help with those projects. So one of the first projects that we did at the very start of the pandemic was put together these COVID-19 datasets. So we were receiving lots of requests for very similar datasets, and so we very quickly put together a set of about 15 to 20 data elements. And because time was of the essence, we were releasing these datasets every day. And as I said, we were getting many requests, and so we kept on adding. We very rapidly kept on adding new and more data elements to every new release every day, and we did these as both an identified and in a de-identified fashion. For those of you who may not be familiar with how to de-identify electronic health record data, this slide gives you a quick summary of it. There's 18 personal health identifiers, and this slide shows you what they are, and this is a part of the process that we follow so that we can make the electronic health record data shareable with researchers. This slide shows some of the elements that we made available both in a de-identified way and in an identified way if researchers had the appropriate institutional review board approval. This shows some of the elements. This included any patient who had COVID, tested positive for COVID, or was suspected of having COVID, and so you could follow the patient journey over each of the data sets as we made each of these data sets available every day. So here there's some, just to give you some sense of some of the projects that use these data sets, looking at social and genetic determinants for COVID-19, modeling the course of care, predictive modeling of patients. I have a slide on that next. So this just gives you some sense of some of the projects. For the COVID 360 project, we came up with a real-time dashboard that combined electronic health record information along with risk factors and the output of a predictive model so that physicians could get a sense of whether their patients might get better or might get worse based on how other patients did with similar backgrounds and similar risk factors. So this was a popular tool that folks were using, especially in the early days. Here's a list of some selected research projects that were using our high-performance computing infrastructure, specifically the machines. And everything from looking at small molecules and doing molecular dynamic simulations to doing genomic analysis and looking at host immune response, and we'll talk about this a little bit more. One of the first things that we helped to discover was that the outbreak in New York City came from Europe, and that it came from a genomic strain in Europe. And we did this by analyzing the viral genomes from the first initial cases and comparing them against the background sequences deposited by other countries. So we got an article in the New York Times for this. Perhaps this plot won't be any surprise to anyone at this point, but we were looking at the positive COVID cases in comparison to a typical flu season. And so as you can see, we had within weeks, we had as many cases as would normally be seen in an entire flu season. Another analysis that was done using our biobank, the blood serum biobank that I mentioned earlier, was trying to understand if there was a group of genes that have similar mutations in it that seem to be correlated with severe COVID. And this work was done using a method called gene burden analysis, and it seemed like that there is a group of genes that had more mutations in it that seem to be associated with worse outcomes. So here's another project that looked at whether Down syndrome patients had more severe disease than other patients matched by age. And in fact, this project found that it is, that these patients had worse outcomes as well, more severe disease. With this project, there was an AI system that found that could improve the detection of patients testing positive for COVID-19 and faster than with PCR tests. So if PCR tests, especially early in the pandemic where they were very more scarce and took a long time to get back, this was a way that we could help to diagnose COVID-19 patients using CT scans. There has been a lot of efforts around drug discovery efforts to help combat COVID-19, looking at the ACE2 disruptors, and here's a project that has worked on this. And lastly, we helped to put together a COVID-19 drug and gene set library, and so these are drug and gene sets that have been contributed by the community, and they're available for anyone to use. And with that, I want to thank you for your attention, and I just want to put in a plug that we have lots of open positions, and you can find them on this careers.mountsinai.org page, or you can reach out to me directly. Thank you.
Okay, thank you, Patricia. It was exactly 10 minutes. So now we are moving to Q&A session. Now let me give priority to the online platform, the SC21 hub. I don't see any questions so far. So. Yes, okay, I should mute my, I apologize if that was too loud. Okay, so I think I'd like to start with like one item that the panelists find most interesting that is an update from what we talked about at the SciM CSC meeting early in the year. So that could be something that you would like to highlight on your from your presentation, or maybe the item that just didn't fit in the format. So I think we're going to keep the same order. So David, if you could go first. The most interesting item since SciM CSC in March.
Okay. Well, actually, what I think is most interesting actually comes in the form of a question, perhaps most directly to Rick, although maybe others will also be able to answer. About 2 weeks ago, Merck got approval for something called molnupiravir, which I think is the first major new therapeutic drug for COVID-19. And I'm wondering, is this one that was identified in your own pipeline? And what is the status of other of the exciting small molecules that came out of your pipeline that went into clinical trials? When can we begin to see other therapies coming onto the market?
Okay, it's I guess the panelists taking over my panel here and asking questions themselves, but that's okay. But
well, I mean, that I think is the most interesting development, but
right.
I want to know how computational
yes,
it was.
So, but Rick,
yeah.
you are next, and since you've
Well,
identified a lot of them,
I can try to answer David's question a little bit. I mean, that molecule was, there was work done at the APS on determining structures with that molecule bound.
Interesting.
So that's an interesting point. That actually was it was a known compound. It wasn't a compound that was created just during the duration of the pandemic. So it's related to other inhibitors that were known. There has been, I think, at least from our effort, there'll be some papers coming out in the next month or so that highlight a couple of drugs that are moving forward in the pipeline and that have achieved more, I would say, clinically relevant concentrations in which they're active. So that's one of the key things. The process of developing drugs takes a long time. The acceleration that happened during the COVID time was really around target discovery, that is the protein that you're going to inhibit, and around the virtual screening. But what we haven't really mastered yet is how to accelerate the series of experimental trials that are required in the process, which requires screening not only for function and antiviral activity, but health and safety issues, PK and so forth, and screening in live animals, 2 different species, at least of one of which is not a rodent, and then getting into clinical trials. So it's that middle bottleneck layer that's between what comes out of our kind of analysis and early experiments and getting into clinical trials that we're still in that time frame. So I think you'll probably see half a dozen or so drugs that will start to trickle onto the market in the next couple of years, or into clinical trials and maybe into the market. What's hitting the market now is basically known stuff. But what we should be enthusiastic about is that this has accelerated the machinery for doing this. To the question about what was the most exciting thing, the fact that once we got our framework put together and we started collaborating more deeply with our AI hardware partners, there's massive enthusiasm for speeding up these pipelines. And I think not only the narrow thing that we were doing, but the broad kinds of things that Daniel talked about, but the ability to actually go deeper into targets for future problems. And so whether it's antivirals or cancer or heart disease or other important areas, I think the same machinery can be applied, right? So I think that's been maybe the most significant kind of secondary outcome of this effort. I'll stop there.
I can buttress that argument a little bit.
Okay, thank you,
I
Rick.
agree that this has accelerated a lot of work. Some of the work we did with the clinical data has already affected clinical practice.
We have a hard time hearing Dan. I don't know if it's on our end or yes, if you could turn it up. I'm sorry, Dan. It's try again, please.
I was going to say I can
That's
buttress
better.
I can buttress that a little bit, and I agree completely with Rick on the acceleration of this sort of science and the resources around it. And we've had some direct clinical applicability in the work we've done in the electronic health records, as well as the mechanistic understanding of the coagulopathies, these coagulation disorders that can have a big impact on COVID-19 patients. And some of those studies led to the direct identification of what are the right anticoagulation therapies to use and the right timing for them. There was a paper that came out at BMJ, and that's directly affected clinical care already in showing much greater efficacy in preventing those coagulation issues with COVID-19 patients. So I think there's the breadth of what we're seeing, as Rick has described, about the ability for discovery of new compounds and reuse of existing compounds, all the way through reuse of existing approved FDA drugs in clinical practice right away, is just such an exciting new perspective on the rate of discovery that we can do with these sorts of resources and affect people's lives now and in the future. And we're seeing that all play out, which is really, really exciting.
Okay, thank you, Dan. How about I look to you, Patricia, since you are very close to the clinical data. I don't know if you were involved in any of the trials that you could tell us about without...
That's a good question. I wasn't so involved in any of the trials directly, but I am involved in where there's been, I think NIH is spending close to $1 billion on long COVID. So all the folks that with the post-COVID syndrome, you know, people who are kind of sick in strange ways. I mean, I think we all know that COVID attacks many systems in the body, and some folks have gotten really tired, and it just have had lots of different problems. And so actually there's a big study that's going on now, and we're helping to recruit 1,000 patients to help study them over the long term. And then of course out of that will be all the data. Of course I'm working on the data piece of it and trying to get that together, and along, as I described a little bit, around the different multimodal data integration, so getting the images and the genomic data and other data together so that we can do some of the analysis. So yeah, I mean, I know some of the trials that we've worked on. I mean, there's a question of how much of the antibody levels, how well do they work? What does it really mean? I mean, of course, all the basic science around this is not totally well understood, and it's still early days to figure out all of these things. So I do want to say, I think the aerosols, I just want to say the aerosol simulations are super cool, if I may say. And those are really nice to watch and try to understand how they're going. So that's all.
Okay, thank you, Patricia. Yes, long COVID is indeed a big problem. I had actually a case close at home, so that's very, very important. So Makoto, you were just complimented. So maybe if you could add something from your side from the simulations, and what do you think was interesting that happened since we last did this?
Yes, there are two things. One is that in these days the aerosol is going to be an important factor for the transmissions, and in that context, people say that partition does not work at all. Mm-hmm. But that is not the case, because sometimes there is some people say that is because the smoke always go beyond the partitions. But when we conduct the simulations, the effect of partition is changing the air directions vertically. So sort of that vertical airflow is going to always reduce the infection risk. So people have said, when people say that that is the reason is the aerosols, and then partition does not work, that is not scientific. The scientific way is always we have to look at more in details physically. There we found out that changing the flows vertically, no matter whether it is across the partitions, but still the infection risk is going to be half. That is the one thing we found. And the second thing we found is that the evaluations of some specific case sometimes is going to be misleading, like one person to one person. So I was showed in my simulations, many people is gathering there. At some places, when we took some countermeasures, some place infection risk increase. But when you look at some specific place, it is going to be misleading. Always we have to look at that as more overall infection risk is going to be very important to think of the countermeasures. These are two things we found.
Okay, yes, very, very interesting. Thank you very much. So now I think we're going to move to, we actually have a question on Hub. I don't know if you can see it. It includes a quite controversial subject, I think. So the actual question is, did your analysis include any drugs like ivermectin that are being used to treat COVID in other countries? So I don't know who would like to go first.
Well, I can say that we included all licensed drugs on the planet, all drugs that are in clinical trials on the planet, all investigative compounds that we know about on the planet, as well as 4 billion other molecules that drug developers, compound makers, and so on had put in their libraries, as well as the ability to generate new molecules. So yes, we included all of that. There were a handful of drugs early on that are licensed that showed pretty good, in virtual screening, pretty good binding affinity to targets, such as nafamostat, for example. Most of the, I would say I don't know how to describe this, the drugs that were mentioned by certain members of the political layer that basically had no impact, though, of course we included everything, and those didn't have any impact. And there was, a lot of, anytime anybody would hear something, we'd go back and look up the screening results. So I think, there was a lot of misinformation, a lot of people out profiteering, a lot of crazy talk. Some of that still goes on, and the science wasn't behind any of that. There were very legitimate activities in the U.S. government and in the agencies asking about known licensed compounds, for repurposing targets, and we definitely gave whatever information we had to those groups as soon as we could. That was very early in the pandemic.
Okay, thank you very much, Rick. I see, Dan, you unmuted yourself, so go ahead, please.
Sure. So we certainly in the EHR data we examined every drug in clinical practice, and actually published specifically a paper on hydroxychloroquine showing that it did not work, and in fact does harm. And we haven't published a paper on ivermectin, but we haven't seen any positive signal from it at all. So it was in the mix and it has not popped up as something that's efficacious. Now it's equally possible that it's simply not being used in the population we're studying, but it certainly didn't for this condition, but it certainly didn't pop up as positive signal.
Okay, so that clearly confirms Rick's findings. I don't know if Patricia, you have anything to add from your data sets.
No, not on these drugs.
Oh, okay.
So, yeah.
Okay, thank you. Okay, so let me check the slide. I don't see any new questions coming up. So maybe we go to something else that we included. So what new COVID-related issues emerge as the active area for HPC to tackle next? So basically looking into the hourglass and the future, what should be focused on? So I guess we're going to go back to you, David.
Okay. I believe that one of the sort of new emphases since the early work was turning, the well-mixed epidemiological models into agent-based models, including, literally billions of individuals interacting in ways that were statistically assembled, or use GPS or cell phone or other data to simulate realistic mixing between people, and not simply at a level of infected or not infected, but having people stratified by the amount of vaccine that they had had and how recently and what kind of vaccine. So these detailed models, I think, are an exciting new prospect. And this actually leads me to another question, which is, we haven't actually used the word vaccine very much in the hour and a half so far. And for many people, that is their number one point of curiosity about the vaccine and how to mitigate it. We've talked about therapeutics that are coming from the computational pipeline, but the vaccines that we have were all developed really very early on, and the only latency was in the testing of them, these mRNA-based vaccines, and then the adenosine-based ones. So I'm curious from the other experts on the panel, have there been major computational contributions on the vaccination side, or is that something that is well in hand without really needing an assist from HPC, unlike the epidemiological side, which clearly is a big data problem of massive proportions?
Well, there certainly are things you can do related to vaccines. So, computational work around antibodies and antibody design is related. Computing, structural dynamics and kind of what is antigenic is part of a series of computational agendas. So the Gordon Bell Award last year, the Romanes Award, with a big team there, they've been aiming at the kind of whole virus simulation, which allows you to get at kind of antigenic surfaces, or what part of the protein and lipid structures in the virus are actually visible. And that could be related to vaccine development. One of the big challenges are that we don't have good computational models of the immune system at the molecular level. We have, epidemiological models, we have kind of process models, but if you wanted to build a virtual test vehicle for virtual vaccine development, we're quite a ways away from that. It'd be like a super grand challenge. It's probably something we should work on. I think the RNA vaccines were more or less designed on whiteboards within a few weeks of having the viral sequence.
Okay, thank you, Rick. I know, Patricia, you had a slide to share.
Yeah, I just was thinking, since we were talking about the next frontiers, and here, let's see if I can, maybe make this easier to see. But I think part of, maybe you can't see, hold on. Let me try again. Can you see now?
Yes, we can see.
Okay. So this slide shows how, as I said, many of the body systems are attacked by COVID, and this slide kind of shows you system by system what gets attacked. And I think, the idea of modeling the immune system sounds very interesting. We know about the cytokine storms and the other kinds of things that happen as the immune response, and there's obviously there's disparities between how men and women and other folks' immune systems respond, and that's a whole area unto itself. But in some sense, this is where talking about getting all the different multimodal data together and using all of it together to help even to just we want to do some multi-scale analysis, right? Just understanding some of the basic science and simulation, say, around the heart systems, and a lot of the DOE labs have done. Long ago there was Livermore did a really nice some of the electrocardio models of the heart, but in some sense, ultimately what you want to do is be able to model all of these systems, right? And from the basic science, but then model it up. You want to be able to do less than real-time simulations of how all of the systems are working together. But COVID is really can really attack many systems at once, and obviously can be quite devastating, and we don't understand. I think a lot of this goes into some of the long COVID, some of the unexplained symptoms that people get with long COVID. So how do the systems interact? How does the virus attack these different things? It's a big, nice big HPC problem, so.
Okay, thank you very much, Patricia. Dan, did you have anything to add?
Are we still on the vaccine question or the broader question?
We are on the what's next for HPC to tackle. What's on the horizon for HPC?
Certainly areas I touched on my talk as I think of horizons we're pointing toward is viral evolution, to understand where the virus is going and how we need to be prepared for that. But of course, this is unfortunately not the last time we're going to be facing this sort of viral invader. So we're starting to focus a lot of attention on understanding the rules of zoonotic spillover events and future pandemics. Because I don't know about any of you, but I certainly never want to go through a COVID-19 event again. So anything we can do to get ahead of that. And the rules of zoonotic spillover are actually quite poorly understood. And it's not surprisingly a massive problem that has global scale requirements. And so we have jumped into that and are pointing a lot of current and future efforts towards that as a way to try, hopefully, a way to help avoid going through this again. But you know, it is nature's feedback loop. We're messing with nature, and nature is messing with us in return, and that's accelerating because the profound degree that we're changing the environment. So I think that's we're of course engaged in trying to deal with COVID-19, but looking towards the future, seeing what's coming, and trying to prevent that, I think, is really crucial.
Yeah, I would agree with Dan. I think one of the key things that the scientific community kind of misses, and this is due to the peculiar structure we have, I think, in separating human health from general biology, is that disease processes in the wild, that is in the ecosystems that are not dominated by humans, of course, co-evolved with those organisms over the billion or millions of years that, depending on what they are. And we need to understand that deeply in order to really understand the total global picture of infectious disease on the planet. And we have barely started studying that, right? Every animal species has a collection of organisms that periodically infect them, viruses, bacteria, fungi, and so forth. And we, in some sense, systematically need to study that across the entire planet. And if you had that picture, and what drives that from environmental, I mean, the kind of stuff that Dan was talking about with these climate change and local environmental changes, how displacement in any of these vector spaces drive that, right? How common is it for viruses to jump species in general? How common is it for viruses to evolve under different virulence or effectivity strategies in general, right? We don't really get that. So in some sense, my view is that what we need to do is stop somehow focusing too much on human. Of course, we have to have care and we have to have that huge investment, but we need to broaden the investment and broaden surveillance and studies and sampling. I mean, there's no reason, now that we can sequence millions and millions of individuals, that we're not sequencing millions of species and their viral hosts and their bacterial hosts to build up a comprehensive picture. We have the computing to do it. We have the AI to understand this. And it's simply a matter of going out and realizing we live on a planet, not in our houses or in our buildings, right? So I think there's a lot of work to be done, and we need to put up, if you imagine you landed on a new planet from scratch and you try to understand the global context, that's how we need to treat infectious disease on Earth, and we're really far away from that.
Yeah, and that's really the One Health framework we're embracing here is you have to do that comprehensively, and genomic surveillance and AI and lots of other, but it's not just that, it's also sociology and human behavior. and land use change, which is economics. It's a holistic view to try to tackle this problem. So we have some funding to get started on that. There's a massive need for much larger scale projects, but I'm always surprised that when we dive into this, how poorly this is understood. And in those wildlife contexts, these viruses and bacteria often aren't disease causing to them. They've co-evolved.
Right.
A good virus is not hurting its host, right? And we're collateral damage in this evolutionary arms race that's gone on in bats and coronaviruses. They co-evolved for millions of years, and they're relatively happy with each other. We're the naive host that hasn't adapted or forced the virus to adapt, and so we're experiencing that collateral damage. And unfortunately, and fortunately, I mean, a 1% mortality rate is not a strong selective pressure for the virus to adapt to do less harm to us. Clinically, sociologically, those are horrific numbers, right? The millions of people that we're losing from this. But in an evolutionary context, that's inconsequential. And we don't want to wait 100,000 years for this evolutionary arms race to even out. We're losing that. We have lost that arms race, and we're losing it against the variants of concern. We actually need better vaccines that are multiplex vaccines, not targeting just one protein. That's just a selective pressure. It already is and will continue to evolve around that. And we need much more sophisticated vaccine targeting, which gets back to simulations and epitope availability. And don't forget T-cell immunity. It doesn't all have to be direct antibody.
Okay, so we actually running quickly out of time, and we have two questions on the hub. So one is, I pasted it in chats. It's monoclonal antibodies are an FDA-approved therapeutic and available in many states. Did your studies play into that? So that's the monoclonal antibodies question. And the other one is anyone doing a serious computational research of mRNA vaccine side effects over time? And there is an example provided known as the mast cells, mastocytosis issues dog by NIH. So who'd like to go first?
Well, our group and other groups at the labs did have worked on antibodies. I'm not aware of a direct connection between, I know of a couple of the pharmaceutical developments of monoclonal antibodies that were happening in parallel. I think the antibody work done at the labs certainly has contributed to the general knowledge about computational design of antibodies, but I'm not aware of a direct linkage between some work done, say, on a supercomputer and the monoclonal antibodies that are available as therapeutics right now. But I think that in the future that could certainly happen as we become better and better at designing antibodies.
Okay, thank you, Rick. Anybody else? Okay. So I think we're just about out of time. We have 1 minute, so let me just close it, since I know some of you have to be rushing to other panels and prepare there. So I'd like to thank very much our panelists for participating and bearing this hybrid virtual and in-person mode. I apologize for anything I was responsible for or I wasn't responsible for. I think in the end it worked out very well. Just for your information, we had about 37 people at some time online. So I think and then we had interesting conversation. So again, thank you very much for participating. And for the live audience, please make sure that you rate the session and let us know and give us feedback of what can we do better the next time we do that. Thank you very much. Bye-bye.
Congratulations.
Thank you very much.
Yeah, awesome work.
Sorry that
Very impressive.
my simulation is a bit different from other guys, so
Oh,
it's
no,
a bit
what-
difficult to involve in the conversation.
No, you were right on your place.
You are the most familiar part of this mitigation. To us, we're deeply impressed. with
Yeah, yeah.
not just the individual detail of the models, like the mesh generation and so forth, but the volume of the cases. It's
Thank you.
huge throughput, really. Thousands of social settings. It's really awesome.